Concepts of Biology 11.3 Evidence of Evolution 11.4 Speciation and 11.5 Common Misconceptions about Evolution. 11.3 By the end of this section, you will be able to explain sources of evidence for evolution, define homologous and vestigial structures. The evidence for evolution is compelling and extensive. Looking at every level of organization in living systems, biologists see the signature of past and present evolution. Darwin dedicated a large portion of his book on the origin of species, identifying patterns in nature that were consistent with evolution, and since Darwin our understanding has become clearer and broader. Fossils. Fossils provide solid evidence that organisms from the past are not the same as those found today. Fossils show a progression of evolution. Scientists determine the age of fossils and categorize them all over the world to determine when the organisms lived relative to each other. The resulting fossil record tells the story of the past and shows the evolution of form over millions of years. For example, highly detailed fossil records have been recovered for sequences of species in the evolution of whales and modern horses. The fossil record of horses in North America is especially rich and may contain transition fossils, those showing intermediate anatomy between earlier and later forms. The fossil record extends back to a dog-like ancestor some 55 million years ago that gave rise to the first horse-like species 55 to 42 million years ago in the genus Eohippus. The series of fossils tracks the change in anatomy resulting from a gradual drying trend that changed the landscape from a forested one to a prairie. Successive fossils show the evolution of teeth shapes and foot and leg anatomy to a grazing habit, with adaptations for escaping predators, for example in species of Mesohippus found from 40 to 30 million years ago. Later species showed gains in size, such as those of Hipparion, which existed from about 23 to 2 million years ago. The fossil record shows several adaptive radiations in the horse lineage, which is now much reduced to only one genus, Equus, with several species. Anatomy and embryology. Another type of evidence for evolution is the presence of structures in organisms that share the same basic form. For example, the bones in the appendages of a human, dog, bird, and whale all share the same overall construction. That similarity results from their origin in the appendages of a common ancestor. Over time, evolution led to changes in the shapes and sizes of these bones in different species, but they have maintained the same overall layout. Evidence of descent from a common ancestor. Scientists call these synonymous parts homologous structures. Some structures exist in organisms that have no apparent function at all and appear to be residual parts from a past ancestor. For example, some snakes have pelvic bones despite having no legs because they descended from reptiles that did have legs. These unused structures without function are called vestigial structures. Other examples of vestigial structures are wings on flightless birds, which may have other functions, leaves on some cacti, traces of pelvic bone in whales, and sightless eyes of cave animals. Another evidence of evolution is the convergence of form in organisms that share similar environments. For example, species of unrelated animals such as the Arctic fox and ptarmigan living in the Arctic region have temporary white coverings during winter to blend with the snow and ice. This similarity occurs not because of common ancestry, indeed, one covering is of fur and the other of feathers, but because of similar selection pressures, the benefit of not being seen by predators. Embryology, the study of the development of the anatomy of an organism to its adult form, also provides evidence of relatedness between now widely divergent groups of organisms. Structures that are absent in some groups often appear in their embryonic forms and disappear by the time the adult or juvenile form is reached. For example, all vertebrate embryos, including humans, exhibit gill slits at some point in their early development. These disappear in the adults of terrestrial groups, but are maintained in adult forms of aquatic groups such as fish and some amphibians. Great ape embryos, including humans, have a tail structure during their development that is lost by the time of birth. The reason embryos of unrelated species are often similar is that mutational changes that affect the organism during embryonic development can cause amplified differences in the adult, even while the embryonic similarities are preserved. Biogeography. The geographic distribution of organisms on the planet follows patterns that are best explained by evolution in conjunction with the movement of tectonic plates over geological time. Broad groups that evolved before the breakup of the supercontinent Pangaea about 200 million years ago are distributed worldwide. Groups that evolved since the breakup appear uniquely in regions of the planet. For example, the unique flora and fauna of northern continents that formed from the supercontinent Laurasia and of the southern continents that formed from the supercontinent Gondwana. The presence of Proteacea in Australia, southern Africa, and South America is best explained by the plant family's presence there prior to the southern supercontinent Gondwana breaking up. The great diversification of the marsupials in Australia and the absence of other mammals reflects that island continent's long isolation. Australia has an abundance of endemic species, species found nowhere else, which is typical of islands whose isolation by expanses of water prevents migration of species to other regions. Over time, these species diverge evolutionarily into new species that look very different from their ancestors that may exist on the mainland. The marsupials of Australia, the finches on the Galapagos, and many species on the Hawaiian Islands are all found nowhere else but on their island, yet display distant relationships to ancestral species on mainlands. Molecular biology. Like anatomical structures, the structures of the molecules of life reflect descent with modification. Evidence of a common ancestor for all of life is reflected in the universality of DNA as the genetic material and of the near universality of the genetic code and the machinery of DNA replication and expression. Fundamental divisions in life between the three domains are reflected in major structural differences in otherwise conservative structures, such as the components of ribosomes and the structures of membranes. In general, the relatedness of groups of organisms is reflected in the similarity of their DNA sequences, exactly the pattern that would be expected from descent and diversification from a common ancestor. DNA sequences have also shed light on some of the mechanisms of evolution. For example, it is clear that the evolution of new functions of proteins commonly occurs after gene duplication events. These duplications are a kind of mutation in which an entire gene is added as an extra copy or many copies in the genome. These duplications allow the free modification of one copy by mutation, selection, and drift, while the second copy continues to produce a functional protein. This allows the original function for the protein to be kept, while evolutionary forces tweak the copy until it functions in a new way. 11.4 Speciation. By the end of this section, you will be able to describe the definition of species and how species are identified as different, explain allopatric and sympatric speciation, describe adaptive radiation. The biological definition of species which works for sexually reproducing organisms is a group of actually or potentially interbreeding individuals. According to this definition, one species is distinguished from another by the possibility of matings between individuals from each species to produce fertile offspring. There are no exceptions to this rule. Many species are similar enough that hybrid offspring are possible and may often occur in nature, but for the majority of species this rule generally holds. In fact, the presence of hybrids between similar species suggests that they may have descended from a single interbreeding species and that the speciation process may not yet be completed. Given the extraordinary diversity of life on the planet, there must be mechanisms for speciation. The formation of two species from one original species. Darwin envisioned this process as a branching event and diagrammed the process in the only illustration found in On the Origin of Species. For speciation to occur, two new populations must be formed from one original population. They must evolve in such a way that it becomes impossible for individuals from the two new populations to interbreed. Biologists have proposed mechanisms by which this could occur that fall into two broad categories. Allopatric speciation, meaning speciation in other homelands, involves a geographic separation of population from a parent species and subsequent evolution. Sympatric speciation, meaning speciation in the same homeland, involves speciation occurring within a parent species while remaining in one location. Biologists think of speciation events as the splitting of one ancestral species into two descent species. There is no reason why there might not be more than two species formed at one time, except that it is less likely, and such multiple events can also be conceptualized as single splits occurring close in time. Speciation through geographic separation. A geographically continuous population has a gene pool that is relatively homogeneous. Gene flow, the movement of alleles across the range of the species, is relatively free because individuals can move and then mate with individuals in their new location. Thus, the frequency of an allele at one end of a distribution will be similar to the frequency of the allele at the other end. When populations become geographically discontinuous, that free flow of alleles is prevented. When that separation lasts for a period of time, the two populations are able to evolve along different trajectories. Thus, their allele frequencies at numerous genetic loci gradually become more and more different as new alleles independently arise by mutation in each population. Typically, environmental conditions such as climate, resources, predators, and competitors for the two populations will differ, causing natural selection to favor divergent adaptations in each group. Different histories of genetic drift enhanced because the populations are smaller than the parent population, which will also lead to divergence. Given enough time, the genetic and phenotypic divergence between populations will likely affect characters that influence reproduction enough that were individuals of the two populations brought together. Mating would be less likely, or if a mating occurred, offspring would be non-viable or infertile. Many types of diverging characters may affect the reproductive isolation inability to breed of the two populations. These mechanisms of reproductive isolation can be divided into zygotic mechanisms, that those that operate before fertilization, and post-zygotic mechanisms, those that operate after fertilization. Prezygotic mechanisms include traits that allow the individuals to find each other, such as the timing of mating, sensitivity to pheromones, or choice of mating sites. If individuals are able to encounter each other, character divergence may prevent courtship rituals from leading to a mating, either because female preferences have changed or male behaviors have changed. Physiological changes may interfere with successful fertilization if mating is able to occur. Post-zygotic mechanisms include genetic incompatibilities that prevent proper development of the offspring, or if the offspring live, they may be unable to produce viable gametes themselves, as in the example of the mule, the infertile offspring of a female horse and a male donkey. If the two isolated populations are brought back together, and the hybrid offspring that form from matings between individuals of two populations have lower survivorship or reduced fertility, then selection will favor individuals that are able to discriminate between potential mates of their own population and the other population. This selection will enhance the reproductive isolation. Isolation of populations leading to allopatric speciation can occur in a variety of ways, from a river forming a new branch, erosion forming a new valley, or a group of organisms traveling to a new location without the ability to return, such as seeds floating over the ocean to an island. The nature of geographic separation necessary to isolate populations depends entirely on the biology of the organism and its potential for dispersal. If two flying insect populations took up residence in separate nearby valleys, chances are that individuals from each population would fly back and forth, continuing gene flow. However, if two rodent populations became divided by the formation of a new lake, continued gene flow would be unlikely. Therefore, speciation would be likely. Biologists group allopatric processes into two categories. If a few members of a species move to a new geographical area, this is called dispersal. If a natural situation arises to physically divide organisms, this is called vicariance. Scientists have documented numerous cases of allopatric speciation taking place. For example, along the west coast of the United States, two separate subspecies of spotted owls exist. The northern spotted owl has genetic and phenotypic differences from its close relative, the Mexican spotted owl, which lives in the south. The cause of their initial separation is not clear, but it may have been caused by the glaciers of the Ice Age dividing an initial population into two. Additionally, scientists have found that the further the distance between two groups that once were the same species, the more likely for speciation to take place. This seems logical because as the distance increases, the various environmental factors would likely have less in common than locations in close proximity. Consider the two owls. In the north, the climate is cooler than in the south. The other types of organisms in each ecosystem differ, as do their behaviors and habits. Also, the hunting habits and prey choices of the owls in the south vary from the northern ones. These variances can lead to evolved differences in the owls, and over time speciation will likely occur unless gene flow between the populations is restored. In some cases, a population of a species disperses throughout an area, and each finds a distinct niche or isolated habitat. Over time, the varied demands of their new lifestyles lead to multiple speciation events originating from a single species, which is called adaptive radiation. From one point of origin, many adaptations evolve, causing the species to radiate into several new ones. Island archipelagos, like the Hawaiian Islands, provide an ideal context for adaptive radiation events because water surrounds each island, which leads to geographical isolation for many organisms. The Hawaiian honeycreeper illustrates one example of adaptive radiation. From a single species, called the founder species, numerous species have evolved, including the 8 shown in Figure 11.16. Notice the difference in the species beaks in Figure 11.16. Change in the genetic variation of beaks in response to natural selection based on specific food sources in each new habitat led to evolution of a different beak suited to the specific food source. The fruit and seed-eating birds have thicker, stronger beaks, which are suited to break hard nuts. The nectar-eating birds have long beaks to dip into flowers to reach their nectar. The insect-eating birds have beaks like swords, appropriate for stabbing and impaling insects. Darwin's finches are another well-studied example of adaptive radiation in an archipelago. Speciation without geographic separation. Can divergence occur if no physical barriers are in place to separate individuals who continue to live and reproduce in the same habitat? The number of mechanisms for sympatric speciation have been proposed and studied. One form of sympatric speciation can begin with a chromosomal error during meiosis or the formation of a hybrid individual with too many chromosomes. Polyploidy is a condition in which a cell or organism has an extra set or sets of chromosomes. Scientists have identified two main types of polyploidy that can lead to reproductive isolation of an individual in the polyploid state. In some cases, a polyploid individual will have two or more complete sets of chromosomes from its own species in a condition called autopolyploidy. The prefix "auto" means "self", so the term means multiple chromosomes from one's own species. Polyploidy results from an error in meiosis, in which all of the chromosomes move into one cell instead of separating. For example, if a plant species with 2n=6 produces autopolyploid gametes that are also diploid, 2n=6 when they should be n=3. The gametes now have twice as many chromosomes as they should have. These new gametes will be incompatible with the normal gametes produced by this plant species, but they could either self-pollinate or reproduce with other autopolyploid plants with gametes having the same diploid number. In this way, sympatric speciation can occur quickly by forming offspring with 4n, called a tetraploid. These individuals would immediately be able to reproduce only with those of this new kind and not those of the ancestral species. The other form of polyploidy occurs when individuals of two different species reproduce to form a viable offspring, called an allopolyploid. The prefix "allo" means other, recalled from allopatric. Therefore, an allopolyploid occurs when gametes from two different species combine. Figure 11.18 illustrates one possible way an allopolyploidy can form. Notice how it takes 2 generations, or 2 reproductive acts, before the viable fertile hybrid results. The cultivated forms of wheat, cotton, and tobacco plants are all allopolyploids. Although polyploidy occurs occasionally in animals, most chromosomal abnormalities in animals are lethal. It takes place most commonly in plants. Scientists have discovered more than one-half of all plant species studied relate back to a species evolved through polyploidy. Sympatric speciation may also take place in other ways other than polyploidy. For example, Imagine a species of fish that lived in a lake. As the population grew, competition for food also grew. Under pressure to find food, suppose that a group of these fish had the genetic flexibility to rediscover and feed off another resource that was unused by the other fish. What if this new food source was found at a different depth of the lake? Over time, those feeding on this second food source would interact more with each other than other fish. Therefore, they would breed together as well. Offspring of these fish would likely behave as their parents and feed and live in the same area, keeping them separate from the original population. If this group of fish continued to remain separate from the first population, eventually sympatric speciation would occur as more genetic differences accumulated between them. This scenario does play out in nature, as do others that lead to reproductive isolation. One such place is Lake Victoria in Africa, famous for its sympatric speciation of chelid fish. Researchers have found hundreds of sympatric speciation events in these fish, which have not only happened in great number, but also over a short period of time. Figure 11.19 shows this type of speciation among a chelid fish population in Nicaragua. In this local two types of cichlids live in the same geographic location. However, they have come to have different morphologies that allow them to eat various food sources. Finally, a well-documented example of ongoing sympatric speciation occurred in the apple maggot fly. I'm not going to pronounce that. Which arose as an isolated population sometime after the introduction of the apple into North America. The native population of flies fed on hawthorn species and is host specific. It only infests hawthorn trees. Importantly, it also uses the trees as a location to meet for mating. It is hypothesized that either through mutation or a behavioral mistake, flies jumped hosts and met and mated in apple trees, subsequently laying their eggs in apple fruit. The offspring matured and kept their preference for the apple trees, effectively dividing the original population into two new populations separated by host species, not by geography. The host jump took place in the 19th century, but there are now measurable differences between the two populations of fly. It seems likely that host specificity okay, I'm gonna skip that word of parasites in general is a common cause of sympatric speciation. That was the end of 11.4. Concepts of Biology, Section 11.5, Common Misconceptions about Evolution. By the end of this section, you will be able to identify common misconceptions about evolution, identify common criticisms of evolution. Although the theory of evolution initially generated some controversy, by 20 years after the publication of On the Origin of Species, it was almost universally accepted by biologists, particularly younger biologists. Nevertheless, the theory of evolution is a difficult concept, and misconceptions about how it works uh, abound. In addition, there are those that reject it as an explanation for the diversity of life. Evolution is just a theory. Critics of the theory of evolution dismiss its importance by purposefully confounding the everyday usage of the word theory with the way scientists use the word. In science, a theory is understood to be a concept that has been extensively tested and supported over time. We have a theory of the atom, a theory of gravity, and the theory of relativity, each of which describes what scientists understand to be facts about the world. In the same way, the theory of evolution describes facts about the living world. As such, a theory in science has survived significant efforts to discredit it by scientists, who are naturally skeptical. While theories can sometimes be overturned or revised, this does not lessen their weight but simply reflects the constantly evolving state of scientific knowledge. In contrast, a theory, a common vernacular, means a guess or suggested explanation for something. This meaning is more akin to the concept of a hypothesis used by scientists, which is a tentative explanation for something that is proposed to either be supported or disproved. When critics of evolution say evolution is just a theory, they are implying that there is little evidence supporting it and that it is still in the process of being rigorously tested. This is a miscategorization. If this were the case, geneticist Theodosius Dostoevsky would not have said that nothing in biology makes sense except in the light of evolution. Individuals evolve. An individual is born with the genes it has. These do not change as the individual ages. Therefore, an individual cannot evolve or adapt through natural selection. Evolution is the change in genetic composition of a population over time, specifically over generations, resulting from differential reproduction of individuals with certain alleles. Individuals do change over their lifetime, but this is called development. It involves changes programmed by the set of genes the individual acquired at birth in coordination with the individual's environment. When thinking about the evolution of a characteristic, it is probably best to think about the change of the average value of the characteristic in a population over time. For example, when natural selection leads to bill size change in medium ground finches in the Galapagos, this does not mean that individual bills on the finches are changing. If one measures the average bill size among all individuals in the population at one time, and then measures the average bill size in the population several years later, after there has been a strong selective pressure, this average value may be different as a result of evolution. Although some individuals may survive from the first time to the second, those individuals will still have the same bill size. However, there may be enough new individuals with different bill sizes to change the average bill size. Evolution explains the origin of life. It is a common misunderstanding that evolution includes an explanation of life's origins. Conversely, some of the theory's critics complain that it cannot explain the origin of life. The theory does not try to explain the origin of life. The theory of evolution explains how populations change over time and how life diversifies the origin of species. It does not shed light on the beginnings of life, including the origins of the first cells, which is how life is defined. The mechanisms of the origin of life on Earth are a particularly difficult problem because it occurred a very long time ago, over a very long time, and presumably just occurred once. Importantly, biologists believe that the presence of life on Earth precludes the possibility that the events that led to life on Earth can be repeated because the intermediate stages would immediately become food for existing living things. The early stages of life included the formation of organic molecules such as carbohydrates, amino acids, or nucleotides. If these were formed from inorganic precursors today, they would simply be broken down by living cells. The early stages of life also probably included more complex aggregations of molecules into enclosed structures with an internal environment, a boundary layer of some form, and the external environment. Such structures, if they were formed now, would be quickly consumed or broken down by living organisms. However, one mechanism of inheritance was in place in the form of a molecule like DNA or RNA, either within a cell or within a pre-cell. These entities would be subject to the principle of natural selection. More effective reproducers would increase in frequency at the expense of inefficient reproducers. So while evolution does not explain the origin of life, it may have something to say about some of the processes operating once pre-living entities acquired certain properties. Organisms evolve on purpose. Statements such as organisms evolve in response to a change in an environment are quite common. There are two easy misunderstandings possible with such a statement. First of all, the statement must not be understood to mean that individual organisms evolve, as was discussed above. The statement is shorthand for a population evolves in response to a changing environment. However, a second misunderstanding may arise by interpreting the statement to mean that the evolution is somehow intentional. A changed environment results in some individuals in the population, those with particular phenotypes, benefiting and therefore producing proportionately more offspring than other phenotypes. This results in change in the population if the characters are genetically determined. It is also important to understand that the variation that natural selection works on is already in a population and does not arise in response to an environmental change. For example, applying antibiotics to a population of bacteria will, over time, select for a population of bacteria that are resistant to antibiotics. The resistance, which is caused by a gene, did not arise by mutation because of the application of the antibiotic. The gene for resistance was already present in the gene pool of the bacteria, likely at a low frequency. The antibiotic, which kills the bacterial cells without the resistance gene, strongly selects for individuals that are resistant, since these would be the only ones that survived and divided. Experiments that demonstrated that mutations for antibiotic resistance do not arise as a result of antibiotic application. In a larger sense, evolution is also not goal-directed. Species do not become better over time. They simply track their changing environment with adaptations that maximize their reproduction in a particular environment at a particular time. Evolution has no goal of making faster, bigger, more complex, or even smarter species. This kind of language is common in popular literature. Certain organisms, ourselves included, are described as the pinnacle of evolution or perfected by evolution. What characteristics evolve in a species are a function of the variation present and the environment, both of which are consistently changing in a non-directional way. What trait is fit in one environment at one time may be fatal at some point in the future. This holds equally well for a species of insect as it does the human species. Evolution is controversial among scientists. The theory of evolution was controversial when it was first proposed in 1859, yet within 20 years, virtually every working biologist had accepted evolution as the explanation for the diversity of life. The rate of acceptance was extraordinarily rapid, partly because Darwin had amassed an impressive body of evidence. The early controversies involved both scientific arguments against the theory and the arguments of religious leaders. It was the arguments of the biologists that were resolved after a short time, while the arguments of religious leaders have persisted to this day. The theory of evolution replaced the predominant theory at the time that species had all been specially created within relatively recent history. Despite the prevalence of this theory, it was becoming increasingly clear to naturalists during the 19th century that it could no longer explain many observations of geology and the living world. The persuasiveness of the theory of evolution to these naturalists lay in its ability to explain these phenomena. It continues to hold extraordinary explanatory power to this day. Its continued rejection by some religious leaders results from its replacement of special creation, a tenet of their religious belief. These leaders cannot accept the replacement of special creation by a mechanistic process that includes the actions of a deity as an explanation for the diversity of life, including the origins of the human species. It should be noted, however, that most of the major denominations in the United States have statements supporting the acceptance of evidence of evolution as compatible with their theologies. The nature of arguments against evolution by religious leaders has evolved over time. One current argument is that the theory is still controversial among biologists. This claim is simply not true. The number of working scientists who reject the theory of evolution or question its validity and say so is small. A Pew Research poll in 2009 found that 97% of the 2,500 scientists polled believe species evolve. The support for the theory is reflected in signed statements from many scientific societies such as the American Association of the Advancement of Science, which includes working scientists as members. Many of the scientists that reject or question the theory of evolution are non-biologists, such as engineers, physicians, and chemists. There are no experimental results or research programs that contradict this theory. There are no papers published in the peer-reviewed scientific journals that appear to refute the theory. The latter observation might be considered a consequence of suppression of dissent, but must be remembered that scientists are skeptics and that there is a long history of published reports that challenged scientific orthodoxy in unpopular ways. Examples include the endosymbiotic theory of eukaryotic origins and the theory of group selection, the microbial cause of stomach ulcers, and the asteroid impact theory of the Cretaceous extinction, and the theory of plate tectonics. Research with evidence and ideas with scientific merit are considered by the scientific community. Research that does not meet these standards is rejected. Other theories should be taught. A common argument from some religious leaders is that alternative theories to evolution should be taught in public schools. Critics of evolution use this strategy to create uncertainty about the validity of the theory without offering actual evidence. In fact, there are no viable alternative scientific theories to evolution. The last such theory, proposed by Lamarck in the 19th century, was replaced by the theory of natural selection. The single exception was a research program in the Soviet Union based on Lamarck's theory during the early 20th century that set the country's agricultural research back decades. Special creation is not a viable alternative scientific theory because it is not a scientific theory, since it relies on an untestable explanation. Intelligent design, despite the claims of its proponents, is also not a scientific explanation. This is because intelligent design posits the existence of an unknown designer by living organisms and their systems. Whether the designer is unknown or supernatural, it is a cause that cannot be measured. Therefore, it is not a scientific explanation. There are two reasons not to teach non-scientific theories. First, these explanations for the diversity of life lack scientific usefulness because they do not and cannot give rise to research programs that promote our understanding of the natural world. Experiments cannot test non-material explanations for natural phenomenon. For this reason, teaching these explanations as science in public schools is not in the public interest. Second, in the United States, it is illegal to teach them as science because the U.S. Supreme Court and lower courts have ruled that the teaching of religious belief, such as special creation or intelligent design, violates the Establishment Clause of the First Amendment of the U.S. Constitution, which prohibits government sponsorship of a particular religion. The theory of evolution and science in general is, by definition, silent on the existence or non-existence of the spiritual world. Science is only able to study and know the material. Individual biologists have sometimes been vocal atheists. But it is equally true that there are many deeply religious biologists. Nothing in biology preludes the existence of a God. Indeed, biology is a science that has nothing to say about it. The individual biologist is free to reconcile her or his personal and scientific knowledge as they see fit. End of chapter 11.